There we go. All right. All right. So today we are going to move on and look at the slightly more complex proposition of understanding eukaryotic gene expression. While things worked pretty nicely with prokaryotes, with operons really don't exist for eukaryotes. There is a far more complex set of mechanisms by which the uh, transcription and translation and expression of eukaryotic genes is controlled. And we're going to talk through a number of those examples today. So while the list of different mechanisms might be quite long, we don't need a uh, understanding down to the very minutiae of each one, but rather to get a general understanding or overview of the different things that could possibly be involved. So, similar to what we looked at yesterday, the big question is how are genes turned on and off in eukaryotes? How do the cells with the same genes differentiate to perform completely different functions? So this is related to the question that I answered. I believe it was, number 11 from the transcription assignment about how do cells that have the same genes end up transcribing and translating different proteins. So the idea that most eukaryotes are multicellular and they needed to evolve to maintain a constant internal environment and face external changes, maintain homeostasis. Um, so the coordination that's required to do all of these things results in genes being switched on and off throughout the life cycle of an organism and from one set of internal or external conditions to the other. There are many, many, many different points of control or places in the trip from gene to protein that can be modulated to change the expression of a gene. And we're going to talk specifically about these 7 different points here, kind of that accompanies the picture on the right. So you can see on the picture on the right the 7 steps listed below, or the 7 points of control listed below. These are just different places where where the cell can uh, affect the expression of a gene before it becomes a fully fledged protein. So if we start at the beginning here with DNA, the first thing we need to remember is that DNA is packed to fit into every cell. If we took the DNA in each cell and stretched it out in one long string, it would be about a meter in length. And we need to pack that into a cell that is anywhere from 10 to 100 uh, micrometers long, right? Or big. So how is that accomplished? So first DNA is coiled in a double helix, and then kind of when we talked about DNA structure here in the first semester, we talked about how they're wound around histones to further package it, kind of in these pictures. So how it's all wound up uh, and looped to form chromosomes. So the first idea that we need to remember, the first content piece that we need to recall is that DNA is tightly wound in a double helix and wound around nucleosomes, so it can be packed together really tightly. Here is another picture on the top. The DNA double helix and 8 of them to form histone. And you can see spiral of the double helix around those histones to form the chromosome in the picture here. Okay, so the main idea is that a group. It's kind of like um, and acetylating histones, so these protein complex, unwinds the DNA. So if a histone is acetylated, it causes the DNA that is wrapped around it to become looser, which enables transcription and turns it on. Right? So that acetylation causes a shape or a conformational change in the histone protein, which allows transcription factors to be able to get in there and bind on it a lot more easily. So while methylation turns off transcription, acetylation will turn it on. So these are the ways, methylation and acetylation primarily, of being able to alter packaging, histone winding of DNA to limit or encourage transcription. We can also control the initiation of transcription through the control of the promoter. We can, or transcription factors, the rate at which they're created and they bind to the promoter can be altered, as well as the presence or absence of enhancers, which can increase or decrease the rate of transcription factor recruitment and therefore the rate at which transcription takes place. So a simple model for how enhancers work, at the top. So here is a more complex picture of the transcription complex. We don't need to memorize what all of these things are, but notice the activators bound on there that are attached to the enhancer sites, which help the recruitment of RNA polymerase and other transcription factors to expedite the transcription of that given gene. We've looked at this a little bit yesterday, but post-transcriptional control is also important, so after transcription. And I gave the example of alternate splicing, so we can have a variable splicing of exons and introns to create a whole family of proteins from the same pre-mRNA transcript. Here at the top is our DNA, and here's our RNA transcript. Depending on what we cut out and what we put in as introns, we mature. Part of this has to do with RNA interference, which I have new up there, is a really promising present branch of research. So RNA interference has to do with what's called siRNAs, which are small interfering RNAs. They're short RNA segments that are between like 21 and 30 bases, and they bind specifically to mRNA that create a section of double-stranded RNA. This double-stranded RNA is a trigger for this cell's own defense mechanisms to destroy that section of RNA. That has to do with the fact that mainly double-stranded RNA is present in viruses and pathogens, and so your bodies have developed defense mechanisms to target and destroy double-stranded RNAs. Right? So these small interfering RNAs can target and expedite the natural degradation of mRNAs to limit the amount of translational copies of the polypeptide sequence that's being made. What this can cause is what we call silencing of a gene, a post-transcriptional control where a gene is turned off even though mRNA is being made, because siRNAs are destroying the mRNA transcript before proteins are even able to be made. And this is, like I said, a very promising new current branch of research, because the idea is to make or engineer siRNAs that could target deleterious or unwanted segments of mRNA, so mRNAs that we know might result in the proliferation of a cancer or of a congenital condition, and we could actually target those potential mRNAs with siRNA and have the body's own defense systems destroy it so that that condition or that ailment is never expressed. That's the genetic engineering or the synthetic engineering of siRNAs. So here is another picture, just to go through kind of how siRNAs are working. So here is an siRNA that is binding with an mRNA before its translation. The siRNA then recruits the body's own defense system, this enzyme called dicer, that chops up the mRNA and degrades it, and so the gene is functionally turned off. We can even control the expression of a given gene after it's been translated or during translation. So we can block the initiation of the translation stage. So there are regulatory proteins attached to the 5' end of mRNA. Like I said, the 5' G cap is really important for the binding of the mRNA to the small subunit. So these regulatory proteins can be activated to prevent the attachment of the ribosomal subunits and the initiator tRNA, so it would block the translation of mRNA to protein. So if on our 5' end, here is where we naturally have our 5 cap, we can block the activation of that binding, hydrolysis of the GTP 5' cap, and we can block translation initiation from occurring. And then last, 6 and 7, protein processing and degradation. So even when we have a finished protein, it needs to be folded correctly and cleaved. It needs to add some other functional groups to target it for transport to the correct area. Certain enzymes, so ubiquitin and proteasomes, are responsible for tagging and degrading proteins that aren't needed or are malfunctioning. And so even after a protein is made, they can be targeted and destroyed using ubiquitin and proteasomes before they're able to be recruited to the area where they would perform their function and having any effect on the organism. So a proteasome is a protein degrading machine. It's basically the cell's garbage disposal, and it breaks down proteins into like small strands of 7 to 9 amino acids that can then be used and recycled to make other proteins. And usually, um, ubiquitin, which is this chemical complex, is kind of the tag that designates a given protein to go to the proteasome and be broken into pieces. So I know that was probably a lot, but my hope is that we have a general idea of different steps that can be taken, different points of control to regulate the expression of eukaryotic genes, even with the absence of a operon like in prokaryotes. Right? So I think something like this picture or the one analogous at the beginning of the slideshow is the best to go over and talk through on yourself, just kind of naming some things that might be part of each of the numbers. And then for a little bit more detailed information, go back through the subsequent slides to look at each in more specificity. So that's kind of where we're going to leave off. We're going to spend a lot of time looking at more examples and fleshing them out here when we go through this in class next time.